Thanks, Matt. That was great. I'm going to switch gears a little bit and tell you how we detect the virus in some of our testing activities right here at Montana State. And I'm going to pick up there on that spike protein that Matt spoke about earlier. In this Scooby-Doo like cartoon here, these green proteins protruding from this virus, you can see that interacting with the host cell. And that spike protein is why the vaccine targets, but it's also important for entry into the cell. And I'm just wanted to remind myself, if I can advance my slide here. There we go. Remember, coronaviruses came, or at least have gone between humans and animals for quite some time. And so this one came out of the blue at us because it's probably the first introduction into humans, at least on this scale. So it was really new, it was novel, and it took us by surprise a bit and got us into a tight spot. And around about July, the state reached out to MSU to see whether we could increase testing capacity throughout our region. And we were happy to help that cause. We put together a team pretty quick and were able to get to work with the CDC protocols on establishing testing standards right here on campus. Now, the CDC originally brought this test out that looked for the genome of the virus. And this technique that we use is called PCR, or quantitative PCR to be more precise, to actually detect this virus. If you want to know more about that, I put a link here. But the CDC really is the governmental agency that drives pandemic responses in our nation. And so this development of this test was not done lightly. It was very rigorously made, but it was very quickly implemented around the country. And so we got a hold of that protocol and began doing that here. Now, I already mentioned this, but I just want to reiterate. This test looks for the virus genome. It doesn't look necessarily for live viruses. And because of that, we don't really know if everywhere that we detect it, it's actually going to be infectious. And that's good somewhat, because we want to err on the side of caution. We want to know whether it's there or the presence of the virus is there. And that's going to help us create strategies like public health rules that keep people safe. And I do also want to mention that this is the gold standard way to diagnose this disease, is by looking for the virus with this technique. There was a lot of, I don't know if everybody remembers, but back when you went in for a COVID test, say over the summer, there was this really painful procedure where they would basically shove a swab up your nose and get way back in the back and cause you to sneeze and gag and everything else. And that was problematic, not only because it was invasive and it kind of hurt, but caused people to sneeze. And we learned very quickly that this thing is airborne, and that's how it gets around. So it causes a lot of issues in that. We still use nasal swabs, but we don't go all the way back anymore. We tend to just get the middle part of the nose, and that's proven to be pretty effective. So qPCR, looking for the viral genome, that's the gold standard. Now, you've also might have heard about these antigen tests. Antigen testing, or antigens, refer to a foreign substance in the body, so something that's not normally there, and it's our immune systems go and surveil for these things. They look for them and they clear them out. And we can capitalize on that biology by making molecules that recognize antigens. We actually have cells in our bodies called B cells or plasma cells that generate a lot of antibodies, and they're very specific when they do that. So if we know what we're looking for, we can generate these antibodies to look for them. And there's companies out there that began making antigen tests and rapidly deploying those out. And there's more information about antigen testing on this website at CDC. I did want to put this table up just to illustrate a few points. Although qPCR, like I said, is the gold standard, antigen testing became very popular. And there's good reason for that. It's generally less expensive. but the turnaround time to get the result is much shorter. Okay. The problem is that it's not quite as accurate. And so there's a balance here. There's a trade-off. And as Matt was talking about, through this pandemic, we've learned a lot about trade-offs and risks and how to weigh risks and what factors are more important. And the interesting thing about our society is depending on which community you live in and which societal factors you value, that influences a lot of how you weigh risk. And so while some parts of the country, like ours, were just fine doing the RT-qPCR testing, a lot of other places around the country switched over to the antigen-based testing. And they had reasons for that. And those reasons weren't necessarily right or wrong. They were just the values of that community. So I did want to talk a little bit about logistics. So one of the other things I would love for everyone to consider is, why not test everybody all the time, everywhere, until we can figure this thing out? And that just has to do, I think, with logistics and how we get things done in a way that meets federal guidelines for diagnostic testing and gives people the right result. And that all starts with our public health system. And in Montana, we have the Department of Public Health and Human Services that's up in Helena. And they kind of get their marching orders from the governor's office and our state government. But they're also taking guidance from the CDC and the federal guidelines. Okay, so these are the folks that try to coordinate vaccine responses, diagnostic responses, responses to any infectious disease in a very concerted way that is consistent with the way that the rest of the nation deals with these things. Now, we get our samples here at MSU from DPHHS, we test them, and we send back their results. At that point, the results get entered into our state database, and then those results are reported to local departments of health. At that point, contact tracing is implemented, so our public health practitioners can go out and try to identify outbreaks or folks at high risk of spreading the disease, and do certain protective measures like isolation and quarantine. Okay, so there's a lot of logistics here. By the time we get the sample, it takes us, well, at the peak of our production, when we were doing about 2,000 a day, it takes 24 hours at least to return results back. So that's even after those were collected and had to be sent to Helena, had to be sent here. And then after we send our results up, it takes a while to process and get things through the database and get the results reported in a consistent way. And so the time between when you get sampled and your test is delivered, your test result is delivered to the local public health is at least 2-3 days. And it's really hard to cut that down anymore just on our infrastructure. The other thing I wanted to talk about, these are some pictures from our lab. These pictures are just meant to illustrate that there's a lot of stuff that we need. We need a lot of equipment, and we need a lot of supplies and reagents to run our tests. And nationally, through this pandemic, there was a shortage of almost everything you can imagine. There were shortages all the way along our production line, the biggest one being this right here. These are called pipette tips, and these carry the sample from a test tube into our testing platform, into our instruments. And the world has gone through a shortage of these things all over, not just in the US, but all over the place. Another shortage were tubes, and we get tubes of any shape, size. I swear, if you have a tube in your drawers or anything at home that's made of Tupperware, we've probably seen it in our lab. So it's been a real challenge to keep up with these things and to stock just enough tubes to sample everyone over time. I did want to throw this up. I'm pretty proud of this. This is our production at Montana State for our diagnostic testing. On the y-axis here, you see the number of tests that we performed. And then this is on sort of a daily level. Okay, so at one point we were doing up around or just over 2,000 tests a day. And you can see we got pretty consistent numbers every day. But over here, you see a bunch of spread of these circles. That just means the number of tests that we do every day is kind of variable, and it's coming down in most cases. That red line is our average, or sorry, yeah, is our weekly average of the number of tests. So we're doing pretty good when we're not having to test a bunch of folks. The bigger news, though, is this positivity rate has plummeted. So of the tests that we get, fewer and fewer of those are positive. And that's what we want to see. And I just have to end here by sharing the names of everybody in the lab that makes this thing work. Just supported by a heck of a team. And these are the most amazing people in my life.